Hi, I'm doing my project on the effects of acute stress on problem solving. So the first study that I looked at was conducted by Duan et al. In 2020. So the background for this study is that acute stress can impair core executive functions such as cognitive flexibility, inhibition, and working memory, which are necessary for problem solving. Stress affects the sympathetic nervous system and the HPA axis. So there's two different types of problem solving. Convergent problem solving requires subjects to derive a single correct solution, and performance is gauged in terms of response accuracy and speed. In contrast, divergent problem solving requires subjects to think of as many potential solutions as possible within a fixed period of time. So with this information, the researchers hypothesized that acute stress will impair creative problem solving, particularly by reducing cognitive flexibility, and these effects may differ between convergent and divergent tasks. So these participants were 70 healthy males with an average age of 19.24, and they were randomly assigned to one of two conditions, either acute stress or control. So all participants were first given a demographics questionnaire to determine their positive and negative affect, and that's known as PANIS. An initial salivary cortisol sample was taken, and then participants either completed a stress task, which was the Trier social stress test, and in this, participants had to conduct a difficult presentation and compute some mental math on a cold audience, or they completed a control task, where they had to just talk by themselves without anyone else in the room and do really easy mental math in front of no one else. Then a second salivary sample was taken. So after the second salivary sample, they counterbalanced divergent and convergent tasks. So they had half the participants do convergent first, and then the other half do divergent first. So in the divergent task, that was the AUT, so participants had to generate as many uses as possible for 3 common household objects. This was scored for fluency, flexibility, and originality. And then the convergent task was an updated CCRAT, and that was 3 different word problems with a point scoring system. After this, participants were given another salivary sample and then a second PANIS test. Additional salivary samples and heart rate were measured in 10-minute intervals. And then they took another positive and negative affect test. So it was found that participants had faster heart rates and higher cortisol levels in the stress group compared to the control group. So that's just showing that, yes, indeed, the Trier stress test worked. And then in the convergent task, stress induction was associated with lower accuracy and faster reaction time. In the divergent task, stress was associated with reduced flexibility to respond with a variety of unrelated answers. So this chart from the research is just showing the difference in the salivary cortisol between the stress group and the control group. So we can see that the stress group is significantly higher. This is before they did the 2 tasks, and then this is after. These are minutes here on the bottom. So this is another figure, and this is showing that between in the divergent task, which was looked at for fluency, flexibility, and originality, only a significant difference was found in the flexibility of the task between the stress and the control group. So in convergent problem solving, flexibility can be manifested only in response timing and accuracy because there's only one solution per problem. In divergent problem solving, there are many solutions, so flexibility is manifested in the variety of responses. Stress induced faster but less accurate responses in convergent problem solving and reduced the flexibility of solution production in divergent problem solving. So the next study that I looked at was conducted by Nair et al. In 2020. So some background for this study is that research has shown that stress impairs creativity and flexible thinking. High levels of creativity have been shown to be associated with increased functional connectivity. Some individuals have a genetic susceptibility to stress, and that can increase an individual's response to stress exposure. So I just wanted to talk about this genetic susceptibility. So here on the right, we can see that this is individuals with a short allele, which is going to be referred to as the S allele of the serotonin transporter. So we can see that they have less of the serotonin transporters here. Yep, see I put a little arrow. And so that's going to decrease the amount of serotonin reuptake, and that's going to make individuals more prone to depression and more susceptible to stress. So the researchers sought to answer how stress affects functional connectivity and language processing regions of the brain. They additionally explored how gender and the presence of the stress-susceptible short allele of the serotonin transporter influenced this effect. Their hypothesis was that functional connectivity between the inferior frontal gyrus, left medial frontal gyrus, left middle temporal gyrus, and right superior temporal gyrus would be affected by stress. They thought that this effect would be greatest in individuals with at least one copy of the S allele, and that the effect would be greater in females. The total participants were 45 healthy participants with an average age of 19.6, ranging 28 females and 17 males. They were then genotyped to determine whether or not they had this copy of the short S allele. So there were two separate sessions. Participants underwent a version of the Montreal imaging test or a no-stress control task. Both tasks were interleaved with task blocks where participants performed an association test that used both convergent and divergent thinking. They examined the relationship between stress and performance and stress and connectivity of language processing. There was no main effect of stress on functional connectivity. Changes in the middle temporal gyrus connectivity with stress correlated with more task solutions for individuals with the S allele, but not with the L allele. There was no significant change in the mean number of problems correctly solved under stress and no stress conditions, and there was no significant difference between the stress and control. So one significant finding that the researchers did see is that gender appears to impact the effect of the serotonin transporter gene on functional activation. Stress reactivity in individuals with the S allele may have a different impact on the functional network integration in males compared to females. So we can see that from the plot here. So these bars are showing functional connectivity strength between the left inferior frontal gyrus and the left middle temporal gyrus. So we can see that there's a significant difference here and here in males and females with the S and the L allele in the stress condition. This suggests that neural responses to stress for processes involved in creative thinking can vary with the influence of genetics. The discovery of such differences might serve towards the development of biomarkers for enhanced cognitive susceptibility to stress. Overall, both research articles suggest that stress impairs divergent problem solving and creativity. Genetics may influence how big of an impact stress has on an individual's problem solving. The size of the impact by genetics may differ between males and females.